and I'm going to turn on the recording for this session. Give me 1 second. Okay. So good morning, good afternoon, and good evening, based on your time zone. Thank you very much for joining today's webinar. We are very fortunate to have Dr. Umer Hassan with us. He's going to talk about a topic which is very interesting. Everybody is wondering about all the pandemic issues and health issues and all that, so it's very relevant today. Global health, the need, challenges, and solutions. And he will share with us what is global health, why do we care, and what role STEM can play to address global health challenges, and how sensors can be used to achieve health quality, health equity, and how to build one. This talk is a result of a collaboration between STEM Matters and Young Professional Society. STEM Matters, the vision of our organization is to enable students to innovate, invent, and deliver quality solutions to contemporary challenges. And our mission is to inspire students to excel in the areas of science, technology, engineering, and environment matters by nurturing their critical and analytical thinking skills. Our objectives are to nurture highly functioning teams to work together and create scientifically sound, innovative solutions to the world's complex problems, create a community of young innovators trained in problem-solving skills, develop high-level scientific programs to provide an international forum for scientific contact to learn, collaborate, and compete for meritorious outcomes, empower students with the skill sets, for example, research, programming, leadership, and others to make them competitive in the higher education and marketplace. And we want to involve underprivileged, give me 1 second, let me mute some of the folks. Sorry about that. And our one of the last objectives is to involve underprivileged youth in science, technology, engineering, and environment matters. Our sister organization, based off of UK, Young Professional Society, YPS, the mission of YPS is to set up and to help support and change the lives of youth by providing personal and professional development opportunities. It aims to bring to the forefront a more progressive, accomplished, and enlightened younger generation of people who will be the representatives and leaders of their communities and who will contribute positively towards the wider society. So our speaker, Dr. Umer Hassan, is a professor at Rutgers University, very accomplished person. He's a winner of Early Career Investigator Award, BMES Career Development Award, Baxter Young Investigator and Emerging Engineer Award, and COSAD New Venture Competition and NSF ICORP Awards. So we have a very, very accomplished person with us. So thank you, Dr. Hassan, for joining. We will enjoy your talk today, so over to you.
All right, thank you so much, Shakil, for the very nice introduction, and thank you so much for the invitation as well. I'm really glad to be here, really excited, and especially excited to, I guess, meet with the students and especially to answer their questions. So I deliberately made my talk not very long and not very technical. So just to provide a bigger picture of the contents for the students, and I would really love to answer any questions and do more of a discussion towards the end on the topics that we're going to talk about today. Can I share my screen? It's disabled.
Okay, go ahead.
All right. Okay, perfect. Okay, so all of you can see my screen now?
Yes.
Okay, great. Okay, great. So today I'm going to talk about global health, the need, challenges, and the solutions. Once again, I really tailored this talk around the students and not the experts. So I would really, at the same time, I'm going to talk about some of the technical content as well, but I try to minimize it a lot so that students can get motivated and understand most of the content, and then they will, and we can have a kind of a very robust discussions towards the end of it. So today I'm going to talk about global health, the need, the challenges, and the solutions. I have been asked to give a very quick recap of my academic journey. So I'm going to give a very, very quick recap. Sorry. Okay. So I was born and raised in Pakistan, Lahore, Pakistan. I did my undergraduate studies in electrical engineering from University of Engineering and Technology, Lahore. In 2011, I came to United States. I did my master's in electrical and computer engineering from University of Illinois Urbana-Champaign. Then I did my PhD from there in electrical and computer and engineering. And most of my graduate work is very interdisciplinary in nature. And you're going to also realize that from my talk today that it has to do with electronics, it has to do with nanotechnology, it has to do with the biomedical engineering, it has to do with microfluidics. So it's really kind of emerging and a collaboration in a lot of different areas, bridging together all of this expertise to develop something new in the sensors. So that was my PhD. After my PhD, I stayed at Urbana-Champaign for a few more years. I was a research scientist there in the Department of Bioengineering. During that time, I was a research affiliate as well at Carl Foundation Hospital in their biomedical research centers. So we were, at that point in time, we were doing this clinical studies on some of the sensors that I developed during my graduate studies. We were validating those sensors with the clinical samples, with the patient samples. And we're gonna talk more about that today. After 2018, I became an assistant professor at the Department of Electrical and Computer Engineering at Rutgers University. I was also a core faculty member at Rutgers Global Health Institute. So this is where I am now. So one important thing that I want to mention is that all the stuff that we do, especially in the terms of the biomedical field or biomedical area or healthcare, that it's not about just doing the research or developing the sensors. It's really about translating those sensors towards the patients. Really the clinical translation for the benefit of the society. And considering that, some of the things that I did at Urbana-Champaign during my PhD work, we filed a couple of patents. I was one of the co-founders along with my advisor, Rashid Bashir there, and a postdoc. So we co-founded a company which was at that point in time called as Erythrocyte and now known as Prenosis. So we're going to talk more about all of that later. And I would again encourage the students that if they have any questions, any specific questions related to especially the interdisciplinary areas, the emerging areas, I would be happy to answer. Now I'm going to kind of formally start to talk about my online presentation. Okay, so here is the brief outline of my talk. Today I'm going to talk about what is global health and why should we even care about it. I am going to provide a kind of a very broad picture of what are these biomems and bio nanotechnology is, how do we use this nanotechnology, these sensors, to address those challenges, to address those global health challenges. We are going to talk about what is the future that we want to create and how do we create them. And this is kind of bridging the above two topics, right? How we can use this nanotechnology, electronics, bioelectronics, biomems, and develop solutions for the global health. I will give just one example on how to develop such a sensor, and that sensor is geared towards HIV/AIDS. I will very briefly touch upon this new next kind of the next century or this century buzzword, artificial intelligence, how we can integrate artificial intelligence and the sensors to improve the healthcare and provide a personalization in the healthcare. And finally, I am going to conclude my talk and hopefully we're going to have a good discussion at the end. Okay, so what is global health? This is the definition of the global health that is kind of listed at the Rutgers Global Health Institute webpage. I'm not going to read through all of it. However, there are three important points here. The first point is achieving equity in health. What does that mean? Achieving equity in health really means that to provide the quality healthcare for everybody, irrespective of their economic background, irrespective of their ethnicity. So quality healthcare should be available to every single person, right? Probably you have been hearing about this topic a lot during all of this presidential debates, Obamacare, Trump care, da da da, in the Congress and the Senate debates. So this is kind of very important, achieving health equity across the globe. So when we say, another important, I think, kind of a misconception among a lot of the students especially, is what is global health? The global health does not mean international health. Global health means international health plus the health of the United States. I understand a lot of these participants are also from the UK. So really the global health is about caring or providing the quality healthcare for every single individual across the globe, including the people in the low and middle income countries, including the people in the developed world, like United States and UK. Okay, the second important component of the global health is interdisciplinary collaboration. And today you are going to realize this thing more, that in order to solve these global health challenges, that we won't be able to solve them while living in silos or while working on these specific boundaries. We need very interdisciplinary collaborations. We need, just to develop a sensor, a biosensor, we need expertise from microfluidics, from electronics, from mechanical engineering, from environmental engineering, from clinicians, from physicians. So all of these people need to come together just to develop a sensor. And then how those sensors could be eventually be translated. And to achieve the global health equity, we need policymakers, right? We need legislators. So a lot of people need to come together to achieve a new world, a better world in which we can have a global healthcare equity for every single individual on the planet, including the United States. The third important component is the population-based prevention with individual level clinical care. A lot of technical discussions can go into this third point. However, I am going to focus, I'm going to kind of very briefly touch that there are two important components that, so while developing all of these sensors to achieve a global health equity, we want to do the studies to develop this prevention and the screening at the population based, while making sure that these sensors are geared towards personalized medicine or individualized medicine. What these terms means, this is going to be more clear in the talk later today. Okay, so what is global health? Next, let's move to. Okay, so in terms of global health, I'm going to give you some of the examples, right? And cancers, let's start with cancer. Cancer is one of the very important global health issues. For example, in 2012, there are 14.1 million new cancer cases and 8.2 million new cancer deaths that happened worldwide. And beyond that, and right, it is such a prevalent condition that almost every one of us knows somebody who may have died of cancer. For example, my uncle died of cancer. We know that Senator John McCain, Republican senator, died of cancer. Steve Jobs died of cancer. Right, some famous people too. Joe Biden's, our next president, died of his son died of cancer. So it is such a prevalent condition. And I think when that happened during Obama administration, they launched this Moonshot 2020 project. And they poured in this billions of dollars, try to solve, okay, with the objective that we're going to solve this cancer problem. And I guess we are already in 2020 and the cancer problem is far from solved. There are significant amount of challenges in order to solve the cancer, a lot of challenges. And I can kind of, just talking about the challenges, I can go on for hours and hours talking about how to solve the cancer and what are the challenges why we were not able to solve them. So anyways, it is such a prevalent condition across the globe. And like some other infectious diseases, this is a condition that most of the developed and the developing world faces even more cases as compared to the underdeveloped regions of the world. WHO estimates that by 2035, these figures can go up to 24 million new cases and 14.6 million cancer deaths worldwide. Currently, I will show you the prevalence of in kind of on a world map in the next slide. So currently we have more number of cases related to cancer in the developed world as compared to the underdeveloped regions of the world. However, WHO estimates that over the course of the next decade, this trend is also going to shift from the developed world to the underdeveloping countries of the world. And one of the two main causes are these, for example, population growth and the aging. We have this aging population across the globe. And they are probably the largest contributors to the increase of the number of the cancer cases across the globe. And so one quick thing for the students is, for example, right, so we have this Cancer Day. WHO recognized World Cancer Day is February 4th. So while we have a bunch of these other days, right, we have a Father's Day, Mother's Day, Brother's Day, at the same time, we have these days as well when we, and if we really care about these issues, we can do something about that in those days, right? And it's not about celebrating these days; it's about raising awareness in the people about all of these issues. Okay, so this is the map over here, the world map that shows the prevalence of cancer across the globe. The regions that are more darker in color, cancer is more prevalent here. For example, you can see, okay, where's my mouse? Okay, right, you can see, right? So cancer is most prevalent over here in China, in United States, then it is most prevalent in India, in Russia, in Canada, in Australia. So cancer as compared to this African continent. So we have a lot of the cancer incidents and the cases are more prevalent in the developed world as compared to the underdeveloped regions of the world. But this trend is even shifting now, according to WHO estimates. Let's talk about another global health issue. Household water purification, unhygienic conditions, unsafe drinking water. Unsafe drinking water as of 2020 kills 200 children every single hour. By the time I'm going to finish this talk, 200 kids will die because of the unsafe drinking water. Right? And
okay, it's working now.
Sorry?
You can go ahead, somebody.
Okay. Okay. So 200 kids will die in an hour by the time I'm going to finish my presentation. Right? And this number of kids who are dying per hour are more than the number of kids who died per hour in World War I, World War II, Vietnam War, Afghan War, Iraq War, all of the wars, last century wars combined. We still have more kids dying every single hour, right? So understanding a lot of these global health issues and caring about these issues can probably provide you the motivation and the purpose on for the students, right? What do they want to achieve in life, or what do they want to do in terms of their studies, or yeah, okay. Here are some other statistics. I'm not going to go into all the details, but two important points. So we have 2 billion people who gets infected with parasitic infection because of the lack of hygienic conditions. We have 500,000 children die every year from diarrhea. And from those people, from all of those kids, 280,000 of them die in Africa, 181 in South Asia, 10,000 in East Asia and Pacific, and almost 40,000 in the rest of the world. Right, so these are unsafe drinking water, unhygienic conditions. Some right. So while living in the developed world in the US and the UK, we take all of these things for granted, while kids are dying every single second and a minute in most part of the world. So what we can do to stop all of these things. Let's talk about one more quick disease, infectious disease, HIV. This world map shows the prevalence of HIV across the globe. The regions that are more darker in color here are HIV is more prevalent over there. For example, this region is Sub-Saharan Africa. Almost 70% of the people who are living with HIV/AIDS across the globe are living in Sub-Saharan Africa. Approximately, I think there are almost 35 million people who have an active HIV infection, and 70% of them are living in this region, Sub-Saharan Africa. We're going to talk more about HIV. And by the way, the world's HIV/AIDS Day is December 1st. Now, this map shows you kind of, not the map, this plot shows you the number of people who are infected with HIV. And this is what I was just saying over here. So, right, so we have approximately 35 million people who are living with HIV. And among them in sub-Saharan Africa, on the right side, 70% of these people are living with, 70% of the total HIV infected individuals are living in sub-Saharan Africa. I'm not showing you a lot of other results of HIV, but a lot of the sub-Saharan African countries have put down a lot of efforts towards decreasing the number of the active HIV infections. A lot of preventive measures have been put in place, and these numbers are potentially going down. USAID has done a phenomenal job as well in starting a lot of these different programs to decrease HIV, new HIV cases in those regions of the world. Okay, by the way, you guys, can you guys see my mouse moving here?
Yes.
Okay, perfect. Okay. All right. So just a quick thing, Rutgers Global Health Institute, of which I'm also a core faculty member, they are doing a lot of these great global health efforts across the globe. This was a survey, I guess, that was done in 2017. We have approximately 653 global health projects that are going on in 114 countries. I am also kind of PI in two of these projects, right? So we have a lot of this great efforts going on across the university to solve some of these great healthcare challenges. And if you are interested in finding out what those projects are and where those projects are being done, I have kind of given you this link. You can watch that later. Okay, so let's move to our next topic. So what is the future we are trying to create and why we are trying to create that future? We already touched upon why, just a little bit, talking about a lot of these global health issues, right? So we're gonna talk more about it. Okay, so one of the problems with our healthcare system, the way the healthcare system has currently been set up across the globe, in the United States too, there's a lot of fundamental problems that needs to be solved if we really want to achieve this global health equity and to provide a quality healthcare for every single individual across the globe. For example, here are some statistics. Again, they are geared towards United States. The number of the people who will be greater than 65 years of age in terms of aging population, more cancer cases. The number of the people who will be greater than 65 years of age in the United States will triple by 2030. By 2020, like almost now, in the United States, there is a shortage of 1 million nurses, 130,000 specialists, and 60,000 primary care providers, right? We have a huge shortage of all of these technical expertise that is needed, right? So in terms of the students deciding on what do they want to do, and if they are looking for, of course, they are looking for the potentially in future, the job prospects too, right? So you can see that in the healthcare, we have a huge demand. On the right, you can see the plot. Essentially, this plot just kind of shows you the registered nurses in terms of supply and demand, that we had a 1 million nurses. There's a shortage of around 1 million nurses. If we look at another country, China, I think the statistics are a little bit old, but the point is still valid, that the number of the insured patients in China have risen from 550 million to more than 1 billion now. And the number of hospital beds per 1,000 insured patients in China has reduced from 4.58 to 2.9. And this means that for the same number of individuals, we have less number of beds available. And the Chinese government have also been pushing to get more and more people insured. So the bottom line over here from this specific slide is the way our healthcare has been set up across the globe, including developed regions like United States. It is not adequate to provide quality healthcare for all. So as an engineer, what we can do, right? There's a lot of this legislation, policymakers, a lot of these decisions need to go into place to really fundamentally change healthcare and to provide the quality healthcare for all. And I cannot do all of that stuff, right? Not a single person can do all of that stuff. So what, as an engineer, I can do? What I do is I develop these point of care sensors. What is a point of care sensor? Here are some of the examples of a point of care sensor, right? You have seen most of these sensors. We have a blood gas analyzers, lateral some other lateral flow assays, like a glucose monitors. We just put so the point of care sensor is it's a small disposable sensor that can give you a result in a minute, and you do not need any expertise for that. Actually, one of the hallmark, I should have included a picture here. One of the hallmark sensors, one of the hallmark example of a point of care sensor is a pregnancy strip test. You can go and buy that for like $1. And all you need to do is to put that in the urine sample. It will give you a result in a couple of minutes. And you don't need any expertise to find the answer out. Right, so this is the sensor that we want to develop. Once you go to the physician's office, the physician office does not have to write you a prescription to go to Quest or other facilities to get your blood work done. You should get that answer right away. That will expedite the therapy that you should be provided. That should help providing the better healthcare to every individual. So point of care sensors can be an important part of a solution. It's not an entire solution as we talked about, right? However, at the same time, it can be an important part of a solution. So this is what I do. I develop these point of care sensors. Another quick statistics in terms of biosensors market, if you guys are really interested into pursuing these areas of the field of biosensors, it's one of the emerging areas in terms of if you wanna kind of quantify the finances. The total industry size in 2017 was $17 billion US dollars. And there was an annual growth rate of 8% till 2024. So by 2024, it is expected to reach greater than $29 billion. Among all the biosensors, we have 56% of the biosensors are wearable, and then we have non-wearable sensors are the rest 5.6% of them. If you look at the market share, right, United States shares almost 81% of the market, and then Germany shares a lot. China is rising here as well in terms of the market share. I don't have the numbers here, but that's another important market that is upcoming now. So in terms of future prospects of the market, biosensors can be a very good area to pursue. All right. Okay, so this is the future that we want to create. Yeah, I don't know how many of you have watched the Star Trek movies or the TV series, but if you have, you know this thing which is called as a tricorder that they have on all of their spaceships, and what they do is they kind of scan the person. It's I think we are a kind of a century out, at least a century out to develop this device. But what this device does is it kind of gives you an answer for every single thing. It does your anatomy scans, it gives you any diagnosis. And a lot of people are kind of working towards developing such a sensor that eventually in the future we should be able to do it. So anyways, right, so this is the future that we want to create, and I hope one day we will. Okay, so what we do, and by what we do I mean what my research group does. So for that, I am going to give you this. So this is a very kind of a broader picture of what my research group does. On the top, you can see this is a hallmark. Again, one of the examples of a point of care sensors. For example, this one is for HIV/AIDS, and I'm going to talk more about that sensor, how to develop that sensor, or how we develop that sensor. So what we do is we just put a prick of blood on the device, right? This is a disposable sensor. That disposable sensor gets to a reader. The reader displays the result in a matter of minutes, and then we throw away the sensor, right? So this is another example of a point of care sensor, and this is what we develop. So here are three main research areas of my research group. Again, just to give you a more research-oriented, broader picture of where the field is going. We develop these automated disposable handheld sensors. They require smaller volumes. Those volumes can come from any clinical sample, can be a blood drop or a urine sample or a saliva or even a sweat sample. Could be your tear samples if you are looking at quantifying the eye health. It should provide the results in 5 to 10 minutes. Another important area that we target is something that is called as the immunoengineering. I'm not gonna go into the very technical details over here, but the premise over here is that whenever we get any infections, could be COVID-19, right? could be HIV/AIDS, could be any infections, any bacterial and or viral fungal infection, our body's defense gets into action. And what we do, we develop the sensors to quantify the state of the defense of our body, how active our body is to fight those infections off. And more importantly, how we can engineer, how we can even improve that. And then the component is this personalized predictive system. This is where we integrate artificial intelligence and the sensors that we develop to improve the healthcare, to develop this predictive sensors or predictive healthcare systems, which will predict rather than relying on the manual input of the physicians. They will predict on what's next or what is the right therapy to give it to the patients. Okay, so this is kind of a very broad research overview of what I do in my research group. Okay, so how we can achieve, so over here in this slide, I talked about this HIV sensor and these point of care devices, right? So how we can develop such sensors. In order to develop such sensors, we use nanotechnology, we use biosensing, we use bioelectronics, we use microfluidics, a lot of these technical areas to develop those sensors. So what is the challenge here? So the challenge is really looking at the scale. If you, so you know your hairs, the diameter of the hair, the size of the hair, or the diameter of the hair is around, I don't know, like 20, 30 microns. 1 micron equals 10 to the minus 6 meters. So if we start to go down our blood cells, in our blood sample, we have a bunch of things. We have red blood cells, white blood cell, platelets. Our red blood cells are a few microns, let's say 3 to 5 microns. White blood cells are 10 to 15 microns, 8 to 15 microns. So the white blood cell, a single white blood cell is even half the size of a hair, diameter of a hair. Then if we look at specific pathogens, for example, most of the bacteria are over here from 1 microns to 10 microns. Most of the viruses, if you want to quantify the SARS-CoV-2, right, the size of the SARS-CoV-2 that triggers this, that do this COVID-19 infection is approximately a few hundred nanometers, less than 100 nanometers. If we talk about proteins, again, coming back to the COVID-19, one of the tests that is recommended on whether you get the previous infection or not, or whether your blood has developed antibodies or not. You have been hearing about this serological testing or antibody testing. In order to find out those antibodies, just look at the size scales. The size of a single antibody is around 10 nanometer, let's say. 10 nanometer is how much? Is 3,000 times smaller than the size of your hair, the diameter of your hair. So the challenge is, how do you find those very, 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 very miniature biomolecules? So how do you develop the sensors to find out, to quantify those biomolecules? So this is, again, what we do. So that is why we use this nanotechnology and biosensing principles to develop such sensors. So I'm going to give you an example of this. Here is an overview of any biosensors. And again, this is a very broader picture. On the left, right, we have samples. So we need to start with any sample. It could be a blood sample. It could be a urine sample. It could be a food sample if you want to find out if the food is infected with any pathogens or not. So we start with a sample. Then there should be something else that we want to quantify. For example, if I am doing a viral test, if I want to find out whether my saliva sample has any SARS-CoV-2 or not, that will be the biological element that I am interested in. If I am doing a serological or antibody testing for the COVID-19, I will be interested in finding out the antibodies. If you are quantifying the blood cells or aptamers for different biological applications, then you will find out that you need to quantify those. So we're going to talk about that as one example. The third important component to develop any biosensor is this transducer. Right? So we need a sensing principle. How do we quantify that? So we can use different sensing modalities. One could be electrical sensing, electrochemical sensing, optical sensing, colorimetric sensing, could be magnetic sensing. Many different sensing principles can be used. I'm going to give you one example today. And then finally, once we use any of the sensors, we get some data. We get some output, and then we perform different signal. What we call the data is a signal, and we do signal processing on it. It involves subtracting or taking out the noise in that signal, it may require signal amplification, a bunch of these signal processing. So with this overview of the biosensors, in very quickly, I just want to give you one example of how to develop such a sensor. Right, again, so this is a sensor that we develop for AIDS diagnostics or AIDS management from a drop of blood. Again, you have already seen this map, which shows the prevalence of HIV across the globe. This region, Sub-Saharan Africa, has 70% of the total infected people. 35 million people are infected, and 70% of them are living over here. And only 1 in the 8 people is able to get tested over here in this region. Okay, so very quickly, how does an HIV affects the immune system? Immune system is the defense of a body. So what happens is, as anybody acquires an HIV, that virus starts to kill a very important type of a blood cell called as the CD4 T lymphocytes. So just for easy, you can just assume it to be a specific type of a white blood cell. So it starts to kill this CD4 white blood cell. So as these number of the viruses starts to go up, the CD4 T cell starts to go down. So if we find out, or especially the physicians are interested in finding out 2 things. One is the number of the viruses that are present in your blood sample, and the number of the cells, the CD4 cells. They are also interested in actually in another cell type, CD8. They are, so the number of those CD4 and CD8 cells, because that presents the how active is the defense of your body against those infections. Right, so again, a quick thing, if we have to count those cells, so here's a challenge, right? So in 1 microliter of a blood sample, again, what is 1 microliter? A drop of blood is approximately 30 to 40 microliters, right? 30 to 40 microliters. So 1 microliter is, if you split the drop of a blood in 30 parts and just take 1 part, in that 1 part, there are more than 5 million red blood cells. There are more than 300,000 platelets. There are more than 7,000 white blood cells. And among those white blood cells, these CD4 and CD8 cells, which are needed to quantify for the AIDS management, they are very, very small in number. They are just 8,000. Sorry, they are just 800 to 1,000 in number in that 1 microliter. However, in infected individual, this count even can go down even below, less than 10. So the challenge here is how we can quantify those cells, less than 10 cells in 1 microliter of blood. Out of those millions and millions of these cells, other cells, how we can quantify those very small number of cells. So the one important thing over here that we can see that the size of these cells, interestingly, is different. The platelets are much more smaller, 1 to 2 microns. Red blood cells are 4 to 5 microns, and then the white blood cells are 7 to 15 microns. So if we have a sensing modality or some form of a sensor that can quantify the cells based on the size, then from the signals that we will acquire, we will know which cell is which type. So we're going to talk more about it. Okay, so this is how currently the cell counting is done in the hospitals, even in today in United States and across the world. This instrument that is shown over here on the bottom right here is something that is called as a flow cytometer. And this is what is inside a flow cytometer. I'm not going to go into a lot of these technical details, but fundamentally what is happening over here is we have a blood cell, which is shown over here, a helper T cell, the CD4 cell, and then it expresses specific proteins from your high school biology. I hope you remember. It expresses some proteins on its surface, CD4 proteins, let's say, or a CD8 protein. What we do is we tag different fluorescent molecules. Now, if you remember your high school chemistry, you know that what is the principle of fluorescence? So we tag these fluorescent molecules, fluorescent antibodies to these cells. And once we shine a specific wavelengths to these molecules, right? So they get to the excited state, they come back to the de-excited states, they emit radiation at specific wavelengths, less energy, higher wavelength. So what we do is, so they emit wavelengths at different colors, if you will. And then we have different optical filters, photodetectors to find out those things. So every cell is tagged with different fluorescent molecules to quantify the number. So what is the drawback? This system works pretty well. There's a lot of this manual processing involved. The people in the labs, they have to process your sample. What are the other drawbacks? The other drawbacks are this, that they are very expensive, more than $100,000. A lot of maintenance cost, $10,000 a year, and it requires, again, trained people to operate these instruments. So, right, it cannot be a point of care sensor. And this is what we want to do to develop these point of care sensors that could be used across the globe. Okay, so if we really want to have this sensors for the resource limited settings of the world, then we need a kind of a more portable, robust, and inexpensive method.
We have a few questions also that came up. Maybe
Sure, please.
Address them now or later. How do you want to do that?
Sure, I'm okay. I'm okay with either thing. Sure,
So,
no, if it's related, sure.
Yeah. So there's a question about cancer, that why the prevalence is so high in USA, China, and Russia. Probably the data that you shared.
Right. So I think it. So I think there are probably two things happening. One is just the people doing more cancer screenings here in the developed regions of the world as compared to the underdeveloped regions, right? So I think one of the things that probably has to go with the statistics and the numbers is just with the people who are more aware and who do more testing. So I think fundamentally, in terms of fundamentally, there is nothing in the developed regions of the world, including United States, Russia, and Canada, that makes the people here more susceptible to cancer as compared to the underdeveloped regions of the world. However, I think the total number of cases are more here because people do get more screened here. Even for HIV/AIDS, only 1 out of the 8 people gets tested for HIV/AIDS in sub-Saharan Africa. However, due to a lot of other reasons, the prevalence of HIV is more in sub-Saharan Africa as compared to the developed regions of the world. However, I think the reason for more prevalence of cancer in the developed regions is more screenings. There's a lot of awareness campaigns that are usually done for the people to get these screenings, and these screenings are usually free in most of the healthcare plans, I guess, that are available to the people. Mm-hmm.
So it's like once a wise man once said that you do more testing, you get more infection. Yeah. So apart from that, so do you think those numbers would be about the same for the rest of the world if they were doing?
Yeah, so I think we can reach to some good hypothesis. Probably yes, or probably they might be more, right? But if I look again, go back to the WHO estimates. The WHO is when WHO or estimates that then the number of the people will, or this trend will shift from the developed region to the underdeveloped regions of the world in the coming decade. So I think the one reason that this is happening is just people are getting more and more screened over here. There's a lot of these programs from the USAID and a lot of other funding agencies, including Gates Foundation, who are pumping in more and more money to get all of this testing done. And I think so. One reason is there's nothing fundamentally that is happening now that will increase the number of cases in the underdeveloped regions of the world. I think that this will probably come down to more screenings that will happen over there with the availability of more resources.
Okay. So that probably, there's another question that probably comes in that the data showed that there were so many children dying from contaminated water, and that's another question that came up. So while we are trying to test more about cancer, we already know there are more kids dying of unsafe drinking water.
Right.
What do you say about that?
Yeah. So I think that's a very philosophical question, and how do you go about it? Right. For example, let me so before giving you an answer, let me give you another example, right? So there's a lot of diseases for which there are very less number of patients, and a lot of the companies don't even develop vaccines for that or develop therapeutics for that, and the reason is there is no economic advantage. But then at the government level, they do care about those people, right? And I think it will come down to answering this fundamental question that our decisions should not be made that one life is important over another. And I think that's the bottom line, or at least I cannot make that decision that one life is more important than another.
Yeah, or if we can rephrase it, that 20 people dying of a disease are any less important than 2 million people dying of another disease. So you want to
Yeah,
focus more, but those 20 people are still ignored for their solutions.
exactly, exactly. Yeah,
Return on investment, but that's the global health issue,
yeah,
right?
yeah.
So there was another comment or question by Professor Wakar from Lincoln,
Okay.
Lincoln. So, and that was on the data on the uh, low market share.
Okay.
The question is, why does India have such a low market share in sensors? While we
No, I
hear,
think, yeah. So that was a few years old, but at the same time, that does not have a specific numbers listed there. That does not mention China and or India and or Australia. Those are the emerging markets, and both the Chinese government and the Indian government have put in a lot of efforts towards increasing the market share, especially in the biomedical health and in the sensors. While that slide does not provide any specific numbers, those are emerging markets, both China and India. Okay, sorry, I cannot hear you.
yeah, please continue. So there are a few
Okay,
things.
okay, great. Yes, definitely. Yeah, feel free to ask me questions in the middle. Yeah. Okay. Okay, great. Yes. So so this was the kind of a system that we currently use in the laboratory settings to quantify these blood cells. Again, this is very expensive instruments. A lot of these maintenance cost. A lot of trained people are needed to operate those instruments. So what do we do is, here's an example. Again, I wanted to give all the students just one example and try to minimize the technical details here on how to develop such a sensor. So here's for example, if I have to develop a point of care sensor for the same CD4 cell count, this is how it should look like, right? I should start with a drop of blood. For example, how it is currently being done, if there is a person who wants to get tested for AIDS or HIV/AIDS, and they want to quantify, and the physician is interested in quantifying these cells. So how the test is done? They usually go to a hospital or a diagnostic lab. A phlebotomist comes, draws a blood sample, a tube, 4 ml, I guess 1 or 2, depending on where they are sending. And then they will send it to the people who run those tests on the instruments, and then they will get the results. There's a lot of manual processing involved. So what do we want to get to? We want to get to a point of care device where we start with a drop of blood that goes to the disposable chip. Chip goes to the reader displays the result, and we throw away this chip. So how we can get to this thing, right? So here is the overall, you can say, the schematic or the process flow on how we can do it. And I'm going to try to minimize the technical content here. So just assume all of these are fluidic channels, more precisely microfluidic channels, because we are talking about this micro ranges, microliters rather than milliliters or even higher volumes. So we are talking about very low volumes, microliters. So we infuse, using a pump, 10 microliters of whole blood. That blood has all of these red blood cells, white blood cells, all the types. Then we introduce another buffer, the red blood cell lysis buffer. What this buffer does, it kills all the red blood cells. Why do we want to kill the red blood cells? Right, a couple of slides earlier, I told you that in 1 microliter, there are 5 million red blood cells and only 1,000 CD4 cells. And if we are for the infected patient, they could be as low as 10. So if I want to quantify out of those millions of the cells, out of those million, if I want to very precisely count those 10, I really want to get rid of all these red blood cells. So I introduce a buffer that kills the red blood cells. We are left with white blood cells. Then we have these micro electrodes. These electrodes looks like this. These are metal electrodes. Again, the size dimensions are very, very small. The width of this electrode is 15 microns, less half of the diameter of an hair, and the spacing is also 15 microns. So what happens is, as a blood cell, a white blood cell passes over these electrodes from these electrodes over here, it is going to generate a voltage pulse. And I count those pulses to know how many cells are there. Then I capture, somehow I have this region, which is I call as a cell depletion or a capture chamber, where I capture the cells of interest by this antigen antibody interactions. So we immobilize these Y-shaped antibodies. What these antibodies does is they have a key and lock mechanism to the specific proteins that are expressed on the cell surface, high school biology. So the cells of interest, which are CD4 cells, white blood cells, those white blood cells have a CD4 protein on the surface. And so I immobilize the anti-CD4 antibodies on the surface. Those antibodies capture the CD4 cells, and the rest of the cells again flow through the second counter. We count out all the pulses again. So if we take a difference of the pulses, we know how many cells are captured. And this is how we can find out the CD4 T cell concentration in the sample. Here, I'm going to very quickly show the 3 modules. So what do we, if I have to optimize this device, I want to do 3 things. I want to optimize this lysis process so I can kill all the red blood cells. I want to optimize my counting so I can count all the cells that pass over through this counter and this counter. The third optimization is I want to optimize my sensor, this component of my sensor where I'm capturing the specific cells. So the first thing is this lysis module, killing of the red blood cell module. Erythrocyte is another term for a red blood cell. By the way, this is a handheld chip, right? A very small 3 by 4 centimeters, half of the size of a credit card. And so what happens is, here are these 3 inlets for the blood, for the lysis buffer. Here's another buffer to stop the lysis process. Again, so we kill all the red blood cells here. The cells gets counted over these electrodes. The cell gets captured. CD4 cell gets captured in this capture chamber. We count the cells again, and we take the difference. Here's a video that shows the lysis process. I hope you can see the video. From the middle, we have the whole blood coming in with all the cells and everything there. And from the sides, we have this another reagent, chemical, lysis chemical coming in. And that is interacting constantly with all the cells and killing the red blood cells. Now, as it is moving more and more, you can see that most of those cell concentrations have been gone. So all of those red blood cells were killed. And what we are left is these white blood cells. You can see this small white dots here. These are all single white blood cells. White blood cell is 8 to 15 microns. We can optimize this entire process in around 6 seconds. We can kill all the red blood cells in 6 seconds. Let's talk about the second important module, which was this electrical counter. We have these metal electrodes, and when those cells passes, it generates all of these voltage pulses, right? Now you can see that some of these pulses are smaller and some of these pulses are bigger. We see that, I told you earlier that some of the cells are bigger in size, some of the cells are smaller in size, right? So smaller cells produce smaller pulses, bigger cells produce bigger pulses. And the principle is if we have a bigger cell flowing through a specific channel, dimension of a channel, it is going to disturb more electric field lines. And the impedance over there will increase. So without going a lot of technical details, simply put, a smaller cells produces smaller pulses, bigger cells produces bigger pulses. Here are the examples. Lymphocyte is a type of a white blood cells, produce smaller pulse. Granulocyte, monocyte is another type of a white blood cell, produce bigger pulses. So we can count these pulses if we plot an amplitude histogram, right? So this is a histogram of the peak amplitudes of these pulses. We can look over here, we can see 2 very distinct cell populations. These are lymphocytes, these are granulocyte monocytes, and these are all total number of white blood cells. So we did this clinical study on more than 100 blood samples. We collected the blood sample from a hospital. We ran that blood sample on our counter. We sent the same sample to that big machine. And they provided us that total white blood cell count. So here is the correlation curve. On the y-axis, we have our own cell counts. On the x-axis, we have a counts from the hospital. And we did this correlation curve for more than 100 samples. And we got a very good correlation of around 0.9. If we look at the dynamic range, and all these samples are coming from the patient samples. So very quickly, let's talk about the third part of this device, which is a capture chamber where we are capturing the cells, right? So in this capture chamber, we have these pillars, right? So this is a very small area. So this entire thing is 3 by 4 centimeters. This is around 1.5 to 2.5 centimeters. In this region, I have more than 100,000 pillars, these pillars. These are PDMS pillars. PDMS is a polymer. And the dimensions of this pillars is, this pillar is 40 microns, thickness of, you can say the thickness of a hair. And the spacing in between them is less than the thickness of a hair. So we initially put these Y-shaped antibodies in this entire capture chamber, which gets stuck to all of these pillars. So when the cells of interest shown here by yellow, they flow through this chamber, they get captured. And then we count before and after to know how many cells are captured. Over here, we can also image them after putting fluorescent antibodies and using a fluorescence microscope. You can see those cells captured. So this is a real CD4 white blood cell that is captured in between the pillars. This is a kind of a top-down image. Here's another image of a chamber in which we capture the CD8 cells. Okay, so let me show you a video of a cell being captured in the chamber. Okay, so this is a cell. You can see that is flowing. I'm going to kind of track it. So video is less than a minute, so hopefully it should be quick. Right, so the cell is still flowing. Okay, now this cell ended up getting captured. So video is still running, right? We can also see the video running from this cell. So this is a cell at a different depth of focus, slightly down. We can still see this cell flowing through the chamber. Right, so in this chamber, we initially immobilized CD4 antibodies. So when these cells, which express CD4 protein on the surface, gets captured. Okay, so we did patient studies in which we collected the samples from the patient, HIV-infected patients from Champaign-Urbana Public Health District. We run those samples we send the same samples to the hospital. They give us the results. We run those on our sensor. We got the results. And again, this is our correlation curve. And for the students who don't know what is a correlation curve is, it compares the two methods, right? On the x-axis, I have a method from the hospital. On the y-axis, I have the counts from our biosensor. And this R-square value shows how well the two match. If my R-square value is a perfect 1, this means they match perfectly. However, so 0.92 is pretty close to perfect. Here is for the CD8 cell, the R-square value was again 0.92. Here's another analysis, which is called as a bland element statistical analysis. It also compares the two methods. The bias value of that method shows on average, my sensor was counting 12 cells more than from the hospital. And the bias value for the CD4 cells, it is showing that on average, my sensor was counting 55 cells less than from the hospital that we got from the hospital. Again, this is some of the statistical analysis that we do when we develop a sensor to compare that sensor with the standard control that is currently being used in the hospitals. So with that, this is the final kind of a content slide that I have today. And it is about just to give you a very broader picture of artificial intelligence, the buzzword for the students, right? And how we can use that with the sensors to develop the next generation systems for individualized medicine. So this is the future. This is the hallmark of what we need to do. We need to develop these point of care devices. We need to integrate these point of care sensors with the other patient data and use the artificial intelligence and or the machine learning techniques to develop the predictive systems. And then we need to do more and more clinical studies to constantly improve our predictive system so that we can predict. Rather than relying again on the physician's input, we want to have a future where we have a system in place that can even predict before the patient progresses to the next stage. What is the right therapy to give it to the patient, for example? So this is what we want to achieve. So with that, thank you so much for listening to me, and I would be very happy to answer any questions you may have. Thank you.
Yeah, we do have a few questions.
Right.
I really thank you, Umar, for this very intriguing talk about the background, the paths you have taken, and your passion about global health and solving problems that not only affect us here in USA, but in other parts of the world. So there's been a few questions that have lined up. First one is, what is your general advice to young people who want to enter this field?
Right. Okay, so I think in terms of entering into a field, So I have a more I have a more generic advice in terms of, right. So I think do what you really care about. And I have more like I don't know, a philosophical answer to this question. We spend 1/3 of our life sleeping, 24 hours a day, 8 hours sleep, good sleep, hopefully. Then 16 hours are left. Let's say we spend 8-9 hours working, professional life, and then rest of the 7-8 hours in weekends. Yeah, you can fairly estimate that we spend 1/3 of our life sleeping, 1/3 of our life professionally, 1/3 of our life with the family, friends, movies, games, kids, everything included, right? So that 1/3 of our life, what we do professionally, should have a meaning. That we want to do something in life, and that should not be that should not be just otherwise. And so it's more of a philosophical discussion, that do what you really care about, and then believe in that, and then make an impact in this world. And for me, it was global health, to improve the quality of people's life. And what I am doing is I'm just doing a very small part of it, right? So you are a very small component of a very big picture. So while realizing that, so pick an example. If you care about the global health, do global health. If you want to do global health, I would advise you to. It's a very interdisciplinary topic. Probably you have learned. So I'm kind of referring to the students and not Dr. Samir, right? Okay, so from the global health perspective, if you want to develop these sensors, then it's a very interdisciplinary area. You need knowledge from electrical engineering, mechanical engineering, microfluidics, some biology, right? So pick and choose your field better. If it's, again, find out what you care about. If you care about, or if you get excited about self-driving cars, do that, right? That's a very important component of our lives. If you get to do the self-driving cars, how many accidents can it save, right? It requires artificial intelligence, a bunch of other developing these computational models, more sensors around that area. So I think I'm going to give more of a generic advice that do what you care about, find what you care about, and then go for it.
I think one question, Dr. Hassan, was from one college kid. If she wants to get into this kind of research, what kind of path she should take in the college and
Right.
the
Okay.
major? And
So I think right. So do some STEM engineering. For example, you can do. My background is in electrical and computer engineering, right? So so you can do electrical and computer engineering, mechanical engineering, biomedical engineering. Do what you like. Again, at the same time, there's a lot of interdisciplinary courses that can get offered in any of the departments. Interdisciplinary courses around the areas of, again, microfluidics, bioelectronics, bioelectrical engineering, biosensors. Take those courses, and then do some graduate work with a professor who is working in the area. The department that is most closely aligned with the work that I described is the Department of Biomedical Engineering. Having said that, all of my background is in electrical and computer engineering, so it's okay. Um, uh, so you can pursue any of these paths. If you want to have a complete overlap, do a biomedical engineering. If you want to do something else, it's perfectly fine. You can do electrical and computer engineering or mechanical engineering, and then explore these areas if you are still interested. Yeah, people's interest in all many times over the course of life.
does it have to be just? Can somebody with undergrad get into these fields and make some impact, or it has to be master's followed by PhD?
Yeah, so I think that's a very good question. So again, so when you get into an undergraduate studies, so the objective is to, I guess, teach you the basic fundamental skills that are needed to make an impact, to make a subsequent impact, whether that is in the industry, whether that is in the entrepreneurship, whether that is in the graduate studies. There are, so I think we should probably take it in a more generic way and like this, that I would advise the student to study in the area in which they are interested, and then caring about the issues, and then try to make this impact in whatever they do beyond undergraduate studies, whether it's a graduate life or not.
If I may ask, you, I know you work at Urbana-Champaign. So did you engage any undergrad students in your research?
Yes.
What,
Okay, yeah, that's a good question. Yeah.
what was the process? Did you go after them, or they came after you, or did
Yeah,
you?
most of the so even at Urbana-Champaign or at Rutgers now, most of the undergrad, so it's really a student oriented thing. The students who usually are interested, so as a professor, I get emails every now and then from the undergraduate students who are interested in doing the work with me, right? And usually I get more emails and more students than I could involve them in the research components that I want to. Right. So considering that, I would advise you, there will be very less cases in which professors will go out looking for those students, because they are already receiving so many emails from the interested students. So if you are interested, definitely reach out to the professors that you want to work with. Another smart thing that a lot of high school students have started to do now, and I have seen this trend for the last 10 years, that the high school students during their senior year, 12th grade, they usually reach out to professors even at that level. They reach out to the professors, they go over there, they do work with them. Of course, they kind of do the same graduate level work, but it's more about the exposure, right? And the professors do understand that they're not going to ask the high school students to do the graduate level work. But at the same time, they will provide you the necessary exposure that you need. Yeah. So definitely reach out to the professors at either the high school level or even at the undergraduate level to get that experience.
All right. So there's another interesting question. There are two questions that I can try to package them into one. That what has been your biggest accomplishment? And I would say there is another question. What was the hardest thing to do during your PhD? What was the major challenge in your PhD that you encountered yourself?
Right. Okay. Yeah. So I think very good questions. Okay. So let's start with the second one. What is the major challenge that I encountered during my PhD? So I know probably I have been lucky that I ended up doing my master's and PhD in 4 years from Urbana-Champaign, where the average number of years is like more than 6 or 7, doing both master's and PhD. So in terms of uh, challenges, yeah, I did not. I'm not I'm not the right person to ask this question. I did not face any significant challenges, but let me tell you the
So,
significant things that I learned
so
from the
you're telling me dealing with weekly meetings with Dr. Rakesh Bashir was not challenging enough for you?
yeah. No, no. So I think that was a really valuable learning experience, right? So for me, yeah. So I think I want to answer these questions from the learning experiences rather than the challenges. So the learning experiences that I got from my PhD graduate studies is to really start thinking about the translational aspects. So whatever the research that you do at the graduate level, think about how they will make an impact, how they will go to the public life, how they will eventually be translated to the public, to the society, right? So that was one important component that learning experience that I learned. And of course, it was a challenge because the students, undergraduate students, when they go to the graduate studies, they does not have that kind of a mindset set at that point in time. The second important learning experience was, again, I think it was not just about doing this novel research. The other component was to, and that there's an overlapping with my other comment too, again, with the entrepreneurship, with really, again, the translation aspects of the technology to the public, finding the patents, how to do a startup. I would encourage all the students to learn about these things. They are very, very important things. Whenever you do go and do an undergraduate study, so think about how do you file a provisional patent? How do you file a full patent? Think about entrepreneurship, very important. So this is, I think, in terms of the challenges, or more like the learning experiences that I have. PhD, I think PhD also trains you to think very deeply, and not deeply in terms of the impact or the philosophically, deeply also in the terms of technically. PhD experience will really push you to think very, very deeply in terms of technical details about anything, about the project. So you should better be kind of trained and or be interested in doing that. And I think my PhD experience was very again a valuable experience for me in doing that as well. So sorry, what was your first question?
So, no, I think you have addressed that. What was your biggest accomplishment has been so far?
Right. So I think yeah, yeah.
Translating your research into a product, probably.
Right. So that is yeah.
Uh,
A lot of important components. And I guess eventually having a good life, having kids, is another important accomplishment. And I cannot just say it's like translation of my technology or a company, just a company. So yeah, I think so probably both of them. Yeah.
So having the being accessible to your family when they need, probably in academia life, that's a biggest blessing. While
Yes.
you do the
Yes.
work, but you
Yes.
can do it in your own time stamps and your own whenever you want to deal with it. There's another question which is of course very relevant to the situation of the coronavirus.
Right.
There's a slim we hear that there is a slim chance that the COVID vaccine can be developed by the end of 2020.
Mm-hmm.
There's lots of politics that went into these kind
Right.
of things. How much truth is in it? And can it be developed so soon? And if so, can it be effective in future treating this virus and other variants? I know we were discussing it when we
Right.
slightly before the start of the webinar.
Oh, okay.
Let's see if you can revisit those discussions that we had.
Right. So okay. So let me very quickly then again talk about the important points related to the vaccine. So I think the vaccine development process is usually genetically vaccine development process is a very long process and takes years, right? Because you wanna first you develop the vaccine, all of the scientific component is done, then you test that on the animals, then the human trials comes in, different phases of the human trials comes in. Even after you give a vaccine to a very diversified cohort of people, you want to track those people over many months to even a year to see whether they develop any side effects or not. So by the time the vaccine is generally available to the public, any vaccine, it's a matter of years. Now coming back to the vaccine for the coronavirus, I think so. If you look at all the statements from the companies that are developing, including so I've been following this Johnson & Johnson as well. I think probably nearby New Jersey. Okay, so if you look specifically for the coronavirus, since it's the same family as a flu virus, and we get these flu vaccine or flu shots every single year, so a lot of companies have a lot of experience in developing those vaccines, in developing those flu shots. And since the coronavirus is the same, so a lot of the knowledge that people and the companies have developed over the course of many years will be leveraged and are being leveraged while developing the coronavirus vaccine. So having said that, getting a vaccine in a year, of course, probably not going to happen, but I would expect the vaccine should come out sometime next year. It shouldn't take many years because, again, we do get flu shots every single year. However, for the coronavirus, but again, we cannot make a one-to-one correspondence with the flu with the flu strain and the coronavirus. But at the same time, it's the same class of family, so there's a lot of the knowledge acquired previously that could be leveraged and being leveraged to develop this coronavirus vaccine. My expectations would be sometime next year. At the same time, since our new administration is more geared towards science, so I would expect whenever the administration is ready to, and especially the CDC, NIH, all of these peoples are ready and say, yes, the vaccine is ready. I will believe in them and I will probably take it. No, I will definitely take it, I guess. Yeah, because of all the traveling stuff, I should take it.
So there are a couple of small policy questions again. Is global health also the public health, or are these two things different? And do we have enough funding in this field?
Um, Sorry, the public health and global health are, sorry?
Is global health and public health the same thing?
Okay, yeah, that's a good question. I think in different, it's the way it's put into different contexts. Again, the global health means the health of the people at the global level. It is not an international health. It means global health. Every single country, including the developed regions of the world, including United States, UK, all the developed areas of the world. And when we say public health, it's more of a, yeah, it's more of a terminology that is usually used for the, so in all the discussions, in all the policy discussions, and or in the literature, it is usually used at a much local level. It's about looking at the people at the community level. It's about looking at the people at the population level. So I think there are these some key differences or context differences. Other than that, a public health, or simply put, public health is a part of global health.
Okay, so and that brings us to another idea. So do we have enough funding into this?
Yeah.
Rest of the world. So what do you know about? Are we?
Yeah. So, uh, so. Yeah, I know. So being a professor, I guess we always need more funding, right? Whether it's healthcare or technology or anywhere, right? We definitely need more funding. I think the funding for the NIH, NSF, all of these federal agencies in the United States is been improving. Even during the Trump administration, the funding was still improved, some of the anticipated. You can say anticipated benchmarks for the funding or to develop new institutes or the new centers. I can see an upward trend towards getting more funding. Whether it is now the question whether it is enough or not, of course it is not enough, and we should spend more towards public health, towards global health, towards technology. And technology is an important part of the public health. So towards addressing all of these concerns, of course, we definitely need more funding. And yeah, it's kind of never sufficient. And so one thing that we learn, in especially in the terms of research, and all the students are eventually going to find out, whoever do the graduate studies, or probably the undergraduate studies as well, that whenever we do research, we will never. Yeah, so whenever you are trying to discover something, whenever you are trying to get new knowledge, what you eventually end up getting with less answers and more questions, right? Then you end up answering those questions. So it's kind of an ever expanding thing. And in order to get this knowledge, an ever expanding universe of knowledge, I know probably you need to have an ever expanding funding situation as well, right, to supplement it.
So we have a question from Saudi Arabia, from
Right.
Coulter University as well. So it's regarding the microfluidic devices for cell detection. Have you or others been able to commercialize this? And what are the challenges in bringing these kind of devices in commercialized POC settings, point of care settings?
Right. Very good question. So two things about my microfluidic device. Again, we did a startup a couple of years ago. We had a couple of SBIR phase 1 grants. I think one was from NSF, the other one I'm forgetting. I think it was from DoD. Then after that, we also had done a Series A funding where we acquired around $6 million to develop the first commercial prototype. So the things are in place. However, having said that, whenever I talk about translation of the medical devices to the patient bedside, it's not a matter of year or years. It's a long road, right? And with all the FDA and all the other regulatory considerations that are needed, it takes a while for any of the medical device from the research component to the eventual translation to the patients. It takes many years, so it's a long road in that terms. Yeah.
Okay, so I think there's one more question, and probably we'll wrap up. And this is again more about yourself, and I would put it in
Sure.
the context that do we have any step-by-step kind of course that you would suggest for young kids, high schoolers, that they can?
Sorry, any step-by-step course, right?
No, hold on. Like, what would you think? Maybe if you can talk to yourself 20 years ago when you were in 10th grade, that
Right.
what kind of things they can engage in, like break apart a pulse oximeter, or maybe look at it. Do you have any step-by-step kind of thing that kids can do to increase their interest in your work or STEM generally? I know you come from UET. There are
Right,
many
right,
folks
right,
like me. We all came from universities where we were taught very traditional course of education,
right,
like
right,
electrical engineering,
right,
mechanical engineering.
right.
Now we are in USA and we are dealing with global kind of young generation. What
Right.
do you think kind of activities we can do? And this comes from a parent, I think.
Okay. So let me address the parent first, or from the parent's perspective.
Right.
So I don't know what age that parents' kids are, but I think engaging the kids at the very early stage in the STEM areas is very important. For example, right, in the STEM toys is very important. I had this many discussions with the people who do this kids therapies related to their developmental milestones. And for example, most of the toys we even buy for the kids, right? If we are just buying them a flying helicopter, that's great. They can fly it for like an hour. Versus if we buy them a puzzle, they can also play it for an hour or 30 minutes. But the outcomes will be very different for both of them. So this is just giving one example, right? So I think for the parents, my advice would be so I think some responsibility falls on the parents too. Then, right, that they need to first develop their own interest in the STEM fields. They would do some they would themselves do some homework, find more toys, find something that will spark the interest of their kids in science. Now coming to the kids, let's say a high school student. So do you want, let's say high school students. So there's a lot of, even in the high school, there's a lot of robotics clubs. There's a lot of other clubs that definitely be engaged in those clubs, right? Now there's a lot of different programming languages that high school students can even do even before the high school students. I don't. They can understand the basic programming principles there, and that can help students learn, or the kids learn some of the basic fundamentals of programming. Or it probably will all come down to logic as a course, that developing the logic skills, the rationality, is very important. So one thing that I would advise to both the parents and the students, and even my graduate student and the undergraduate students, like it's probably applicable to everyone, develop critical thinking. You should always be asking why and how, to the parents, sure, but to your professors too, perfectly fine. You should think more deeply about everything, whether it's a working of a machine, whether it's something else, right? So think critically, and once you do that, you will learn more, you will learn new, and you're definitely going to have this phenomenon in which you will ask more questions rather than whatever the number of answers that you were getting. So I think it's an overall way of life, rather than I'm going to give you one thing. It's an overall way of life that the parents and the kids and the teachers and the students, they all need to develop to enter into the future, or for all the challenges that we need to solve for the future. I think, yeah, big challenges: climate change, global healthcare, and in order to solve any of these challenges, there are big hurdles to overcome. Yeah.
Very good. So, Dr. Hassan, just to add to your critical thinking skills development, STEM Matters, the organization that is hosting this webinar, we are trying to institute that. We're trying to help our young kids, middle school and high school, kids who want to do something really interesting, probably as interesting as Dr. Hassan has shared the projects with us today. The STEM Matters, we take kids, we help them develop, sharpen their STEM skills, and we take them to national and international competitions. So we will be starting another cohort, another session in spring 2021. Please see our visit our website for more information. In a week or so, we'll be posting all the information. But definitely, as Dr. Hassan said, critical thinking skills are really, really important in the industry, in the research, almost everywhere, to take care of the huge challenges that the humanity is facing. And if we can be the agents of change in a good way, making positive difference in our societies, that will be mission accomplished. So with that, Dr. Hassan, thank you very, very much for your wonderful talk. Very insightful, very beneficial. You addressed a lot of questions, and we wish you really a blessed day and blessed life. Thank you for your contributions. And we thank our audience for joining us as well. With that, I'll, Dr. Hassan, any last departing comment before I say goodbye? You're on mute.
All right. Okay, great. Yeah. So thank you so much for the invitation. I think I had a very good conversation, discussions. And one thing that I would still encourage, if anybody has still any questions, I would, anyone, like the students from across the globe, right? You are most welcome to send me an email. I would be very happy to answer any question that you have. And probably my kind of a departing comments will be to care about, to have some purpose of life, and to care about something, and then think more deeply about it, and then do something about it. Yeah.
Wonderful. Great. Well, thank you very much, everybody.
Right,
have a good rest of the day and evening.
Have a wonderful time. Thank you. Bye-bye.
Take care. Bye-bye.